You're listening to BioTalk with Rich Bendis, the only podcast focused on the biohealth capital region. Each episode, we'll talk to leaders in the industry to break down the biggest topics happening today in biohealth.
Hi, this is Rich Bendis. I'm your host for BioTalk, and we have a treat for the listeners today. We have a twofer. We have two rather than one, and both are MDs. And that's going to be a stimulating conversation, I think, for all of us. We have a Baltimore-based clinical stage pharmaceutical company, and we have the Chief Medical Officer, Christopher Chapman. Christopher, how are you?
I'm fine, and you?
Great. And then we have Adam Kaplin, who is the Chief Scientific Officer. Adam, good to talk to you again.
Nice to talk to you, Rich.
Great. And normally we do one person, but I think you guys have worked as a team long enough that this is going to go extremely smoothly, and we're going to have very easy transition from one to another as we roll through this interview here. So I've introduced you briefly, but I think the best thing we do is to let each of you do a self-introduction on what you think the listeners would like to know about you. So I'm going to start with Dr. Chapman. And basically, can you talk a little bit about how you integrated your skills from medicine, the military service, and business, and into your current role that you've had, Dr. Chapman?
Yes, thank you, Rich. So I graduated from Georgetown University School of Medicine. I did my internship at Georgetown. I did my residency at Georgetown. I did my fellowship at Georgetown in OB/GYN and pediatric anesthesiology. And my fellowship was as an anesthesiologist. I decided that I was doing a lot of work with anesthesiology. I would go in the morning and pick up all the drugs for the patients to do the cases. And so I would paralyze patients. I would wake them up. I would put them to sleep. So I learned a lot about drugs. So being an anesthesiologist was my interest in the pharmaceutical industry because I knew all the drugs. I knew all the side effects. So I think that was a perfect interest into coming into the pharmaceutical industry about 25 years ago. And then prior to that, I was in the military. I was in the Air Force. I was a B-52 crew chief, and I flew all around the world. And being in the military, I learned about management, leadership, mastering life versus death, and dedication and loyalty and relationships, and how to be on and manage projects. So got most of my structure while I was in the military. And then since I've been training in the pharmaceutical industry for about 25 years, about 2 years ago, I took a course at Harvard Kennedy School in executive education. So that way, with these public companies, I could review the 10Ks, 10Qs, public filings, and budget. And so my whole circle was starting in the military structure, then anesthesiologist in my trade, and then doing business at Harvard University Kennedy School. And that brought my career full circle to where I am now as the president and director and chief medical officer at MyMD Pharmaceuticals.
Fantastic. That's a great and diversified background. And you mentioned Georgetown like 3 times. So we're based in Rockville, Maryland, which is part of the biohealth capital region. You're now in Atlanta, as you mentioned to me, but you know a lot about what's been going on in the Washington, D.C. area. So it's good that you have the familiarity here, Christopher.
Yes, I still have a home in that area too. So I go back and forth between Maryland, offices in Baltimore, but I still go to Georgetown and right up and down the street, the NIH and FDA. So I'm still a local person in that area now.
Very good. And we'll learn more as we find out where you are with the company, but you're going to have a lot more interactions with the FDA as you continue to grow here. So,
yes, no question about that. We look forward to it, Rich.
Yeah, thank you. And also we have Dr. Adam Kaplin, the CSO for MyMed. Adam, you want to give the background to the listeners?
Sure. I mean, I'm a local boy compared to Chris, but I'll try to give you a sense of my background. So I actually have been at Johns Hopkins for the past couple of decades. I did my MD, PhD in neuroscience residency training there, and then joined the faculty. I've retained an adjunct faculty status there, but 3 years ago, I came over and began working full-time with Dr. Chapman, with Chris, and the other members of the team. Essentially, my specialty working at Johns Hopkins was both seeing patients who had mood disorders, particularly related to inflammation. So I studied the way inflammation causes things like depression and how depression causes inflammation and worsens diseases. And along the way, I had the privilege of trying to patent several of the discoveries that I made with my colleagues. And it just became increasingly clear to me that the exercise of trying to get articles up on the shelf when we have literally millions of articles now in thousands and thousands of journals is a useful exercise, but ultimately getting it to the patient is what is the real rewarding thing, coming up with novel treatments. And so I have found that academia is a great place to come up with new ideas, but industry is the place to develop new drugs. So that led to my belief that if you can combine the two, you're in the best possible shape. So now I work with Chris and the other members of MyMD to try to bring this drug through the various stages of research, usually through academic centers, and then take it hopefully along the way of eventually bringing it to the aid of patients if we're fortunate enough.
Right. It's great to see two prestigious institutions between Johns Hopkins and Georgetown, both in our region, have some affiliation with MyMed and your background. So I'm sure that as alumni, they will be glad to hear about your path to potential success with MyMed. So, and by the way, you mind if I call you Chris and Adam?
That's perfect.
That's okay. Great. So let's talk a little bit about the evolution of the company now then, Chris. How did MyMed get started? Who is really the key driver and visionary behind MyMed? And then talk a little bit about how Baltimore got selected as a location for this company.
Okay, Rich, I'll be glad to discuss that. Johnny Williams Sr. is the founder. He's the designer. He's the brainstorm behind the whole operation. And back in 2014, he developed an alkaloid, let's say from mysamine. And we determined at that point that we could take that product and change one carbon on it from a 6-member ring to a 5-member ring. And because of that, it would decrease side effects and we could give it orally. So that's how we got started. So I did all the manufacturing after he designed the drug. I did all the manufacturing, I did all the preclinical, I did all the tox, all the way to phase 2. And so the way the company evolved, we were a small company in Florida in 2014, developing the drug. And then in early 2021, we merged with Acres. Acres was a public company with a shell, but no product. MyMD was a company with a product, but we were not public. So we did a reverse merger. So we merged MyMD, the drug, MyMD1 into Acres, and we came out as the superior company and became MyMD Pharmaceuticals. And that was a reverse merger that took place in 2021. And since that point, we've been running the phase 1, phase 2 for the company.
Was that done through a SPAC, Chris?
No, it wasn't.
It wasn't a SPAC. Okay. So basically you had a shell company that basically had shareholders, had some money, and you had a product and you were looking for money, and it was a perfect marriage.
Rich, I couldn't have said it better. They had a public company, they had the shareholders, and we had a drug. And so we did reverse merger. So MyMD came out as the leader of the company of MyMD with the ticker MyMD, and cash in the bank and shareholders ready to go.
Well, that's not a bad way to do it. You got to the public arena much quicker than most companies do, but there's pluses and minuses about being public. We can talk about that later. We don't have to go into that right now. Adam, let's talk a little bit about your interaction with Chris, how you two came together, what really you saw as the major incentive for you to stay engaged with Hopkins, but actually make this transition into this pharmaceutical world that had no guaranteed success with it. And, there's no tenure in being the CSO of a small public biotech company, is there?
No, there's a. Well, now you tell me that, Rich. Where were you a few years ago?
Now you know.
So let me tell you, where I came in was I was actually fortunate enough to be asked to do some research on this interesting compound, MyMD. One, through my involvement in that capacity, I learned about the research that had already been done at Johns Hopkins by Mario Cattaragli, who is a brilliant pathologist and basic science researcher. Chris and Johnny had been working with him for several years. And what really attracted me, Rich, was the following, which is that Mario had shown that this drug in mice, and he's published these results, slowed the aging process. Now, the reason why that attracted me is I've known for a long time, as part of my education, that if you get rid of all of cancer, for instance, you'll change the life expectancy by 2, maybe 3 years. And you say, well, gee, we invest billions of dollars and cancer is such a major killer. How could it only affect the lifespan by such a short time? And the reason why is as we get older, the risk of cancer goes up, but so does the risk of heart disease and lung disease. And if one of those falls down, there's something right behind it waiting to come in, meaning it is aging that is ultimately the disease, if you will, that causes us to have all of the chronic conditions associated with aging and all of the suffering that we go through. So knowing that and hearing that this drug could slow the aging process, now that doesn't mean it just extended life, because if we all could be 150 years old and demented, that's no fun. Risk of dementia increases with age. Age is the number one risk factor for dementia. So what he had actually shown is that this drug not only extended the lifespan, but preserved the health of the animals in the process. So that was enough for me when Chris and Johnny came to me and showed me Dr. Cataragli's initial results. I was in.
I guess when you say slow the aging process, I'm a little further along than both of you in that, but I hope it's not too late if we can get this drug approved to slow my process down a little bit.
No, it's a great question, a great point. And what Mario had done is he'd actually started when the mice were the human equivalent age of 60, and then went to essentially 100 years of age in the human equivalent of those mice. So he wasn't waiting. He didn't start at birth or anything. He waited till people were past the midway point, if you will.
Well, there's a lot of people of my genre that wish you a lot of success with this drug, for sure. Chris, let's talk a little bit about the relationship there is that exists between inflammation and disease and how that impacts the significance of MyMed and the work you're doing.
Yeah, thanks, Rich. Well, I think that focus on inflammation and disease goes back to inflammatory and autoimmune diseases. And that disease space is growing with significant aging population and unmet medical need right now. So that's why we focused on inflammation. And just to go back and talk about what Dr. Kaplin talked about with Dr. Cataragli's, is one of the first studies he did was Hashimoto's thyroiditis, which is an autoimmune disease. And so we went from Hashimoto's thyroiditis, and then we did an MS study in the mouse to look at the MS in the mouse and in the brain, and then we looked at sarcopenia. So there was all three autoimmune diseases. So that's how we got started with the thyroiditis at Hopkins. And then we noticed that with sarcopenia and Hashimoto's thyroiditis, there are no disease moderating products. Everything is for symptoms. And so we felt that we need something that could actually cure the person instead of just dealing with the symptoms. So that first study at Hopkins and thyroiditis made us realize that was the area we want to focus on, autoimmune disease and inflammation.
That's interesting. And people don't know how these interactions impact the body and bodily functions. And I guess only through the research that you're going through, sometimes at the basic research level, like Hopkins is the leading basic research university in the world, does some of this actually materialize so that it helps give you a path forward for the direction you're going to go with potentially commercializing a product. So, Adam, talk a little bit about how MyMD works, especially in diseases associated with the inflammation, and what does it do that differ from current treatments that are out there today that people claim help?
Great. So no problem. I'll get that done in one set. No, give me a minute to cover some of that.
You can have a couple minutes for that one.
Okay, great. Essentially, what MyMD1 does is it is able to block the production of inflammatory cytokines, particularly TNF alpha. So what does that mean? Well, when the body is invaded by anything, bacteria, fungus, virus, there's an initial immune response, and that is TNF alpha, tumor necrosis factor alpha, goes up. That's what causes us to get a fever and shake, and it then sets off a whole cascade of activating other cytokines. Cyto means cell, kine means kinetic movement, and these are the two immune cells will communicate with one another like two neurons use neurotransmitters. Two immune cells will use cytokines, and TNF alpha is kind of the master regulator. It's the first one to go up. And not surprisingly, given that MyMD has been shown to work in multiple different autoimmune diseases as well as aging in general, that this is a general process that gets going with whatever your favorite autoimmune disease is or aging related condition. So what this is a new version of a drug that is actually the number one selling drug in the world is Humira. These are drugs like Humira are anti-TNF alpha treatments. They bind up and soak up TNF alpha wherever they find it. They don't cross the blood-brain barrier. They're basically large antibodies or large receptors. But what's unique about our drug is for the first time, these anti-TNF alpha drugs that are $40 billion a year, just Humira has been $20 billion a year prior to getting a generic. It still makes a ton of money because there's such a huge need for these illnesses. And what is different about MyMD, one, is unlike these other drugs, which need to be given as an injection or an infusion, ours is oral. It's a small chemical, not a huge antibody. It is selective, meaning it only inhibits the autoimmune component of inflammation, meaning that when you first get an infection, even before your body learns to recognize, gee, this is a particular bacteria to worry about, it has what's called an innate immune response. You don't want to take that out because you need that in order to prevent yourself from getting overwhelming infections. MyMD1 doesn't affect this innate immune system. Instead, it affects the adaptive immune system, the one that gets tricked into thinking the body is foreign and starts attacking the body. So that selectivity means safety to us. And the last thing is it crosses the blood-brain barrier. Also, something about being a small molecule is not only can you be taken orally, it will cross the blood-brain barrier. And I will tell you, I specialize in treating patients, the neuropsychiatric aspects of MS, multiple sclerosis. And you can't use any of these anti-TNF treatments for MS because it turns out that if you block TNF in just the body, not the brain, because you don't get across the brain, you'll actually potentially risk increasing inflammation in the brain. So we have oral availability, we're selective, we cross the blood-brain barrier, and that gives us a real advantage in an area that's very exciting because anti-TNF alpha treatments are the big blockbuster treatments to really help patients with a number of autoimmune diseases.
When you talk about Humira, basically, it's not a bad poster child for you to try to emulate for what you're trying to accomplish in the world. But Chris, let's talk a little bit about sort of the potential indications, the patient populations, and sort of where you are in the clinical process with the FDA right now.
Sure, Rich. So just to go back and so that it's clear, we did all the manufacturing. We did a 1-year dog study, a 1-year rat study. We did 2 phase 1 studies prior to the phase 2. And so everyone understand we have a IND for phase 2 in rheumatoid arthritis if we want to. We have an IND for phase 2 in Hashimoto's thyroiditis if we want to. So that's the background. Now for the study we just completed, the sarcopenia phase 2, we needed the background information, the 2 phase 2 studies and the dog study and the rat study to get to the phase 2. So the phase 2 study was done, and the principal investigator was out of Hopkins, by the way, and he's a very good colleague of Dr. Kaplin's. And so it was a 40-patient study, 32 active and 8 placebo, 28-day study. And when I went to the FDA or spoke with them about the study, they actually picked the biomarkers for the study, and they picked TNF-alpha, TNF-alpha receptor 1, and interleukin-6. So those were the 3 biomarkers for our endpoints. And so the good thing about the study is we sort of knew of a 28-day study that it was an outpatient study. Patients came in on day 1 and they got pre-dose labs. And then for 8 hours, they got PK. So we measured the biomarkers during the 8 hours. And then they went home and came back on day 7, had the same thing done. Then on day 14, 21, 28. So we sort of knew that if we get them a good blood PK on day 1, 7, 14, 21, 28, we may see some efficacy. And I think that's exactly what we saw with our results, where the p-value for the TNF-alpha was 0.008, for thyroid TNF-alpha was 0.002, and for interleukin-6 was 0.03. So we're very satisfied with those results. And so the plan now is we're in a very good position. So we have a phase 2 IND to start rheumatoid arthritis if we want to. And we're putting together the final clinical safety report for the sarcopenia study. That'll be ready by December or something like that. And then we'll present that to the Food and Drug Administration. And when we do that, the FDA will decide on whether we do a long-term phase 3 or what the next steps are. So that's where we are. And we were very satisfied that 2 days after we announced our phase 2 results for sarcopenia, the FDA approved the IND for rheumatoid arthritis. So I think they saw the press release. But anyway, I think we're in a very good position now. So we can start 2 phase 2 studies, one with Hashimoto's thyroiditis, one with rheumatoid arthritis. And with the big picture is see what the FDA decide about the end of phase 2 study for sarcopenia.
When you have multiple indications that your drug might be applicable to, and you're an emerging company that has resources that sometimes can be stretched, how do you decide and prioritize which direction you're going to go when you have the opportunity to go a couple directions, Chris?
Rich, I couldn't ask that question any better. I'm being pushed and pulled. Some shareholders would like for us to go into rheumatoid arthritis, and some say finish sarcopenia. So we're taking a conservative route, and that is we'll present everything to the Food and Drug Administration at the end of the year and decide on whether we continue with sarcopenia. And the thing that Dr. Kaplin mentioned is there's no drug on the market for sarcopenia. There's no medication. There's nothing approved. So it's a wide open space for this company. So let's say if I had to make a choice, I would say we'll stay into that space and maybe do a long-term study and try to get accelerated approval in sarcopenia because there's no competition.
Well, I guess the benefit to it also, and Adam's going to answer a little bit further, is that I know other companies that have been able to have platform technologies, and you have licensing opportunities or strategic partnerships that you can develop with multiple companies if you so choose to go that direction. But Adam, I'd like to get your opinion on this.
Well, the only thing I just wanted to add to what Chris had said is to give him full credit here. He didn't just pull this out of a bag of tricks. He actually saw the opportunity to partner with Charles River and run our drug in their assay, their model of rheumatoid arthritis. And it's a really good model because it basically takes antibodies against joints, injects them into rats. And so you see the inflammation in the joints. By finding the right group to do those experiments, Chris came back and said, oh, by the way, let me show you this. And the data just knocked our socks off. It really showed the best response, better than what Charles River had seen with other anti-TNF alpha treatments. Our drug really just dramatically shut down the inflammation. So that gave Chris the opportunity to say, hey, this is a great place to be, and to get everybody behind the idea of this is an obvious target given power that it has in the animal model.
Yeah, thanks, Adam. You may want to mention that in that study that Dr. Kaplin is talking about, we compared MyMD1 to Embro, and Embro lowered inflammation by 37%, and MyMD1 lowered it by 47%. So we beat them.
Yeah, you'll take 10%, right?
I take it every day.
Let's talk a little bit about Supera-CBD and a little bit about how Supera-CBD functions and who are the potential beneficiaries of that. And we'll start with Adam.
Yeah, so Supera-CBD, the name is loaded. We're sort of suggesting that this is a superior version of CBD. I don't think I'm telling any secrets out of turn here with the name Supera, but essentially it takes CBD, the exact molecule of CBD, and it takes one carbon away, making a 6-member ring into a 5-member ring. And that has made an enormous difference in terms of how this works. So the important thing to understand, the first thing about the difference is that CBD is a cannabinoid, meaning it is found in cannabis, which is marijuana. It's the second most abundant cannabinoid, the first being THC. THC binds in, activates inside of us the CB1 receptor, that's the intoxicating receptor, but the CB2 receptor is the one on all the immune cells. That's the one that it presumably is involved in anti-inflammatory and anti-pain effects. And perhaps there's evidence to say many other things such as anti-epilepsy and the like. So the first thing we knew is that it doesn't affect CB1, which is good because it's not intoxicating, just like CBD is not intoxicating, which is why it's not a scheduled drug. But the other thing we found out is just removing that one carbon made it 8,000 times more potent at activating CB2. Just means that you could use a lower dose and get a potent activation of the CB2 receptor. And again, since everybody knows that CBD is safe and people are taking it in large doses, that if you could deliver something that is much more effective, a much lower dose, that would be good. And then the last thing we know now is that experiments being done back at my mother institution has shown that in a model of inflammatory pain, inflammation is involved in a lot of different types of pain, that this drug very rapidly leads to anti-pain effects in an inflammatory model of pain. And that effect is specific to the heat-induced effects. Heat turns out to be same receptors that do spicy food is involved in heat. So this is called a TRP channel. And these channels are involved in a lot of pain. It doesn't cause numbness, so you wouldn't lose the ability to, if you had pain in your hand, to use your hand, but it does shut down that pain-induced heat sensitivity. And what is really dramatic to us is that in this model, CBD had no effect whatsoever. So we saw as big a difference as you could hope for in the potency at CB2 receptor, this safe receptor involved in inflammation. And two, we actually have now demonstrated with our colleagues who have done the research at Hopkins that this shuts down, significantly ameliorates pain, whereas CBD does nothing in that model. So we are superior in this respect. We're not just boasting with the name.
So Supera-CBD, you're not superior, but it is reflected in what you're classifying it. But I see that Chris has got a comment on this, but also, Chris, after your comment, I'd like to know, does FDA have a special division for anything related to CBD, or does it, since you're dealing, in a pharmaceuticals, does it get into the general pharmaceutical area?
Well, that's a good question. That's what I was going to comment on. So what we did is, Rich, we took the structure of Supera-CBD and took it to the DEA, and the DEA determined that doing all our development, Supera-CBD was a non-controlled substance. In other words, we can do all our control work with it, all the work with it early on, and it's not controlled. So I think that gives us a big advantage. There's a division in the FDA just to deal with CBD and different products like that. So I think we're in pretty good shape having that ranking now.
Yeah, and it's nice to have the DEA blessing, right?
Yes, it is. It's very nice. Adam, you want to say something about the DEA?
The only thing I would add is, and I know I'm putting the cart way ahead of the horse here, but we have shown that we don't have any activity at the CB1 receptor. Because CBD doesn't have any activity at the CB1 receptor, and therefore doesn't cause intoxication of any kind, CBD was able to come off scheduling, and it is a drug that is prescribed. It was developed to treat a rare form of epilepsy in kids. Epidiolex is CBD that's purified as opposed to synthesized, which is what ours is, because ours is a new chemical entity with a patent. But essentially, in my best guess is that since we don't have any effects so far that we can even postulate that are intoxicating, my greatest hope is that we will preserve this lack of scheduling, and certainly don't anticipate this will be a controlled substance in the way that other painkillers currently available, like opiates and the like. This should be much safer.
I think for the listeners, and I'm talking to Christopher Chapman, who's a Dr. Christopher Chapman, President and Director and Chief Medical Officer for MyMD, and Dr. Adam Kaplin, Chief Scientific Officer for MyMD. And we've been talking a lot about the science, but let's talk a little bit about this entrepreneurial journey that you both have been on right now. And this may be something off script a little bit, but I think it's something the listeners are going to be very interested in. And I'd like to know about, as you've been going through this journey, which is not an easy one, what are some of the surprises that you both have been surprised by? And what do you see as some of the greatest challenges that you've overcome, but yet have other major challenges to overcome before you actually successfully enter the market? So this is a question for both of you, because I think you're both focused in a little different area in working with MyMD. So let me start with Chris. Chris, talk about this journey, surprises, and sort of the major challenges in front of you.
That's certainly a good thought for us, Rich. And I think that for me, general toxicology is a major challenge because you develop a drug, you need to go through manufacturing, and before the FDA will give you an IND, you got to do your tox work. And you never know, you're doing a 7-day maximum tolerated dose, doing a 28-day study with the rat and dog. If anything goes wrong, you got to repeat those studies. And everything that can go wrong will go wrong. So don't worry about it, just keep trying. So those are the major challenges. I mean, I'm like up at night making sure that 28-day study works, because unless you get 28-day study, you can't get an IND. That tox is a major challenge for us always. And then once you start the study, once you start a phase 1 or phase 2 or whatever, there's sort of little headaches. And I just give you a good example that I thought about if a question like this ever came up. So for this study we just finished, it was phase 2. We had 39 patients. We need one patient to complete the study. We had a patient lined up to go. They had done their pre-labs and everything was fine. They came in to be dosed. I won't talk about the site they came to, but everybody was waiting to announce the last patient in the study. The patient comes into the study that day and said, I don't want to be in the study. So that set us back 15 to 30 days because we had to get another patient to screen them to get them on schedule. So those are the kind of things that you look at and say, well, that's a clinical trial. But it worked out. We got the last patient, we finished the study, the results were good. And so we're very happy about that.
So normally they say it's generally going to take twice as long and twice as much money. And how true is that for you guys?
It takes twice as long and twice as much money.
Okay, we'll talk about future challenges, Chris, in a second. Adam, what about you as we go through to where you are today and things you've had to overcome, and what do you see as the major hurdles in the future?
Well, I know this is going to come as a complete shock to everybody, but the one thing that I was surprised about naively that is the same in academia and industry is that politics are involved in everything you do. And whether it's trying to get something through the FDA or trying to find the best approach to go forward, there's logistics and politics involved in everything. But what I have been really glad about is that coming from academia, I still maintain that our best chance of making meaningful developments going forward to really help mankind is to use the novel ideas and creativity that's in academia with the know-how of people like Chris to get things through the regulatory and development process, fundraising and stuff. But being a part of it makes a difference. So I'm a neuropsychiatrist, and it turns out that MyMD, one, as we were developing it, had some characteristics that looked to me like it would suggest it would help with depression, inflammatory depression. We don't have time to get into it. And we did experiments, and sure enough, it is an anti-inflammatory that also will treat depression if it's inflammation-induced depression. So you say, okay, gee, that's great, because now we know another aspect of this drug that we wouldn't have known if I hadn't been a psychiatrist, neuropsychiatrist interested in this. But I will tell you, and this is a hint of what's going to be coming down the road, working with my colleagues, all the same people, Dr. Cataregli and the like, we are now learning by studying MyMD more about how antidepressants work in general. You know, I love the give and take. I love the fact that you can still be doing important work that crosses that division between industry and academia, as long as you keep in mind the first principles of what your main goals are.
And I would imagine one of the big differences is in academia, publications and patents are important, but in industry, it has to go beyond the publication and the patent.
Yes.
Yes, you got to get through the FDA.
And actually to the marketplace. So, Chris, you're sort of leading into the next one. We talked a little bit about the challenges. Let's talk about your future goals, milestones, and what are the major hurdles you see going forward, whether it's related to talent, whether it's fundraising, regulatory, clinical, whatever it might be.
Well, with talent, I think we're surrounded by some of the best talent. And one of the things I should have mentioned earlier was in the early part of my career, I was the medical director for Quintiles, which is now Cuvia, which is the largest CRO in the world. So I used to run the medical development for that. And so because of that, for all the projects I use going forward, we use IQVIA. So I'm pretty safe about phase 1, phase 2, phase 3. I think they carry a nice spot for that. And then for the tox work, I think Charles River does a very good job for that. And then with talent, one of the other parts of human talent is because of Dr. Kaplin, I was able to find a very good person out of Hopkins with Dr. Brager. She's executive vice president of drug development. She was trained by Dr. Kaplin at Hopkins. So she fills in a lot of spots for me in drug development. And then I have a good CMC person, a good tox person. My tox person, Gary Wolf, has been with for about 6 years. So I can rely on that kind of talent to make sure I get these projects through. So other challenges, of course, is regulatory. Whenever we finish a phase 2 of whatever, we got to go to the FDA. You may get a 5 minutes with the FDA, you may get a 6 minutes, but it has to be fast. You need your team in order and ready to go. You don't know what the results will be, but I can tell you, the last time I spoke to the FDA I had a project. I have 2 projects, and we had 4 on my side, and they had 16 people from the FDA. And so we went into the meeting, and we were on tense, And so the FDA said, Dr. Chapman, we're going to do this drug like the other drug. And I could have fell out because it's exactly what I wanted, you see. And so things like that, you have good days, and we always worry about when you have a product at the FDA in IND or phase 1 or phase 2 or whatever, to get a call on Friday from the FDA saying either stop the study or move forward. So that Friday before the 30th day is always a really thought for us when we go forward. The other thing is always fundraising. So we go to a lot of meetings. We have to keep cash in the company to sustain it. So that's always a challenge. And we have a pretty good finance team to help us with that part of managing the company.
You did a great job, Chris. I mentioned 4 things. You covered all 4 of them. So if you can respond to the investors in the same way, you're going to have no trouble raising capital.
Thank you very much, Rich.
Adam, you're based in Baltimore and you're right in biohealth capital region. So talk a little bit about how MyMD has really been able to take advantage of the resources you have in your own backyard, whether it be at Hopkins, other resources that you find that are beneficial to you in Maryland or in this region that you've been able to take advantage of?
It's a great question. And obviously, as Chris is mentioning, whether it's getting in a car and driving out to the FDA to meet with them in their offices, whether it's going to NIH, having discussions with them, whether it's being located right across the street with a connecting bridge to the basic science building where research is done at Johns Hopkins Medicine campus. We are in the biotech park right across the street. Very convenient for us to be able to get access to the brilliant collaborators that we have and with whom we're able to consult to get their information. I will tell you that were we not in this particular situation, we might not be here now. And I say that only not because this isn't a great drug and the like, but we've gone through COVID during the evolution of this particular company. And if it wasn't for the perseverance of all involved, and props to Chris for being the head of the pack here to keep us on track. And if we didn't have such good access to the opportunities we have in this area, I think it would have been very difficult to get as far as we have in the course of all of the closures that happened in the first couple of years of our development. So I think that we're very fortunate to have the kind of leadership we have, but also to have the opportunity, as you say, to be in sort of research central where we're located here.
Yeah, I'll just follow up a little bit, Chris, because it sounds like you're on the front line dealing with a lot of existing and potential investors. And a lot of times people in our region, biohealth capital region, get pressure from the investors to move to Boston or San Francisco. And I just wondered if you've had to fend off that much and be able to defend it very easily based on all the resources you have available to you in the Baltimore and the Maryland area.
Yeah, Rich, we've been pretty fortunate because being in Baltimore, we 30 minutes to Washington, D.C. and 2 hours and 2.5 hours in New York. So I think we're pretty good being next to Johns Hopkins. So everybody likes that we're an incubator at Johns Hopkins. So that works out very well for us. All investors like that. And so sometimes they think we are Johns Hopkins. So, and we don't say no.
That works to your advantage most times.
Yeah. And we always say we got a staff member, Dr. Kaplin. So what can they say?
Well, the other thing is, though, then you say, we have a great relationship with the university, but we're industry driven. We want to get to the market. We want to make you money, our product's scalable, and we think we can get a great return for your investors.
Exactly.
I know we've gone on a lot about MyMD, both of your backgrounds, where you are with the science. Is there anything that we didn't cover that either of you feel that the listeners would be interested in knowing? Chris?
Well, I just want to make it very crystal clear, Rich, and you've been in the industry for a long time. You know a lot of biotech companies, and for a company our size to have 3 INDs for 3 phase 2 studies, I think we're in pretty good shape. I feel very good about the company. I mean, I work for a lot of companies that do not have INDs, do not have phase 1, phase 2. So I feel very good about the future of the company because of those aspects.
Yeah, nice thing is you're not a one-trick pony, and if you have to pivot, you have something to pivot on. How about you, Adam? Anything you'd like to close with the listeners?
I would just say, if you had told me even 5 years ago, I'd be working with the likes of the people I'm working with on a drug that slows aging and that is a better widget than the currently number one selling drug, I'd say, well, hooey, there's no way that's going to happen. So the fact that that has happened, I just feel privileged to be able to be in this position.
Well, whenever you get to the human clinicals and you need candidates to help stop aging, let me know. I'll raise my hand. I'll be one of your guinea pigs.
Got you. We'll sign you up.
Yeah, okay, great. I just want to thank both of you, Dr. Christopher Chapman, President, Director, Chief Medical Officer, and Dr. Adam Kaplin, Chief Scientific Officer for MyMD, for being on BioTalk. And we want to come back to you after you continue to go through this clinical process and closer to market or in market and do an update with you. So thank you both for being on BioTalk.
Thank you. We appreciate it and look forward to it.
Thanks, Rich. Thanks for all the guidance and the opportunity.
Thanks for listening to BioTalk with Rich Bendis.